All right. So I'm going to start with a brief regulatory overview of individualized medicines to sort of set the stage for what we're going to discuss. I do want to note that I am focusing on US regulations. So this is the process for how this is submitted in the United States. Some of the other countries are modeling a little bit after us, but other countries also have a little bit more flexibility. So I'll talk about what the individualized patient expanded access IND is and how it is different from an N of 1 IND, the process for submitting one of these applications to the US FDA, and very briefly, a little bit about how to implement these trials. And I'll share some resources if you need additional information. All right, so the US FDA regulations refer to an individual patient expanded access, IND, and this is what we think of most commonly when we hear about expanded access, or sometimes the term compassionate use. That phrase compassionate use isn't actually in the US drug regulations, but that's what we kind of think about here. In order to qualify for those kinds of INDs, there's very specific criteria that must be met. The patient must be in a serious or immediately life-threatening disease or condition. These INDs tend to have a faster than 30-day review time. The FDA can do these within hours. They can do them verbally over the phone. They can happen very quickly depending on the patient's clinical status. And then there's also administrative processes that happen at the institution, again, in order to speed up the timeline here. For these specific individual patient INDs, they need to rely on an existing active IND of an investigational product. So that original IND is where we describe the preclinical experience, the toxicology, how the drug is manufactured, what is going on in the existing clinical trials for that product. So when we think about these regulations for this kind of individual patient, these drugs have to already have been developed and been studied in order to be using this. So this is quite a bit different than what we're talking about for these N of 1 or individualized medicine INDs. So these N of 1 INDs are different because it's a new molecular entity. This has not been studied before. It has not been used before, treated patients, and so on. So therefore, a full IND application is required. The application needs to include any proof of concept, preclinical studies, experiments that you're doing, toxicology studies. The toxicology studies, and we'll talk a little bit about these, aren't necessarily the same full package that you're using when you're doing commercial drug development, but we still need a toxicology study. And we still need to describe how the drug is going to be manufactured and how stability will be measured going forward. So the process for submitting an N of 1 IND application, the FDA strongly recommends using a pre-IND meeting process, and we have always done that with the cases that we have submitted. So the pre-IND meeting process, generally you request a meeting 60 days ahead of when you want the meeting to happen, and you submit your data 30 days before that meeting occurs. Depending on the specific patient and their prognosis, you might be able to ask for a shorter timeline, but it is very, very important that if you're doing that, your data is ready. I know that there's a lot of the patient is declining or needs treatment pretty quickly, and you're still running experiments. So there's a lot of timeline jockeying, but it doesn't do anybody, the FDA or you as an investigator, any good to be asking for this meeting and not have the data readouts ready for that. So I would strongly caution against that. So the package that you have to submit generally has a lot of proof of concept data, a protocol summary or plans for what your toxicology study will look like, which species you're going to use, how many, what the dosing schedule is going to be, what your plan for necropsy, all of that. So they don't have to actually be done. Usually the pre-IND meeting is where you come to alignment on what the toxicology study is going to be. And you also need to describe your plan for the manufacturing of the drug. Which vendors are you going to use? What's the stability going to be? The guidances indicate that you really should be using the clinical batch of drug for your toxicology study. So again, there's a lot of timeline jockeying between when you can get manufacturing slots, when you can get toxicology slots, and so on. The IND application, it will be submitted as a research IND, likely with the physician who is caring for the patient serving as the sponsor. So there's two different kinds. There's the research IND, which is generally what academic medical centers submit, and then the commercial IND for more commercial drug development. So all of the sections are needed because, again, we need to provide the data, the toxicology studies, the manufacturing, and so on. However, because it's a research IND, you do not need to use ECTD, the common technical document. All of the sections generally are needed, but you can tailor them to the individual case. So the sections where you're describing the disease process and what has been done in the literature historically can be very much tailored to that specific case, that specific gene mutation, and so on, and what we might know about the natural history of that disease. Generally, because the FDA is reviewing all of this data, they generally require this 30-day review time. However, it's been our experience that the FDA reviewers are very open to sometimes what we're referring to as a rolling review. So a lot of our applications have been submitted with preliminary toxicology data, but the longer term or the necropsy data or some of the pathology, I know that takes a while to get that to read out. If you can make an arrangement with the FDA and tell them, preliminary pathology is going to be available on this date, and the final report's going to be available on that date. We have had experience that the FDA has been pretty amenable to accepting the toxicology data on a more rolling basis. Again, you have to get that agreed to ahead of time. And then once the IND itself is active, you do need to follow all of the other IND regulations regarding amendments, safety reporting if there are any adverse events, annual reporting if your patient is going to be treated on an ongoing basis. So those regulations are still in effect. Okay. And I say that I'm touching briefly on institutional implementation because as much as the scientific data and generating all of those experiments takes a long time, getting an entire hospital to agree to doing this also takes quite a bit of time. This is becoming more and more popular. So many institutions are requiring specialized internal review. These internal review committees will have scientific review, ethics evaluation, finance. The people paying the bills at the hospital definitely want to get involved. The clinical staff. So there's a lot of internal review and a lot of people to check in with before they'll start accepting doing an N-of-1 trial. Because this is a research IND, full IRB, institutional review board review is required. So they can't use the same administrative processes that individual patients generally use. In triggering full IRB review, that often triggers a lot of other reviews at hospitals, scientific review, certain departments across the hospital, pharmacy, if you're doing MRIs, there's a lot of research-oriented review that happens in a normal clinical trial that also get triggered here. And I have put in bold and all caps because it's the thing that has made this the most successful in the cases I've been involved with is to get your nursing and clinical operations staff involved early. The people who are doing research are very accustomed to industry-sponsored trials. There's a protocol, there's a very rigid way of doing things, and this needs a lot more flexibility and finesse. So when you get these people on your side early, they're able to figure this out. And a lot of times this sort of interfaces with patient care. And sometimes those people aren't as familiar with research. So there's a lot of people learning to do new things. So it's always really important to make sure that, especially for patient safety, that we're getting these people involved early. And then briefly, I'm just going to let you know what the FDA has published. There's 4 guidances. There's one for the non-clinical testing, so what they recommend for toxicology and so on. They are permitting, when it makes sense, single species rodent toxicology studies. There is a guidance about what considerations for chemistry, manufacturing, and controls are needed for these types of drugs. There is a clinical recommendations guidance that recommends what sort of monitoring should be done. And finally, there's an administrative and procedural recommendation. And a lot of what I was talking about today about recommending a pre-IND meeting and what the IND should look like and who should be the sponsor, that's covered in these guidances. And then the InnovOne Collaborative, we have a lot of resources. Roger and I have both participated in several webinars for the InnovOne Collaborative on different topics. We have templates for protocols, consents. There's been a lot of patients who have gone through this. The investigators have given seminars as well on what their experience has been. So all of our work group generated content is posted here. And then I will make another plug for our annual meeting the day before the OTS meeting. And again, Roger and I will be there to chat with anyone else. And at this point, I see Erin coming off on video to see if there's any brief questions before we answer the ever important question, well, how do we pay for this?
Yeah, exactly. So Ashley's given an amazing overview. If anyone has very specific questions, we can take them now. Otherwise, we'll leave time for a broader discussion at the end of Roger's talk. If you have questions, please do feel free to put them in the Q&A. You can put them in at any time during the presentation. It's not disruptive.
I do have one in the meantime. So since I'm online, I think I can ask one. You briefly mentioned the different systems across the world. So I'm based in Europe, and I think so. Do you have any knowledge on the EMA system? Because I think there is no requirement to go to the EMA before doing these N-of-1 therapies in Europe. I'm not an expert, but it is correct.
So I personally have only experience with one case in one country in Europe, and it is my understanding that there is a process called the named patient program, which mirrors a little bit of our individual patient expanded access in the US, where a physician needs to obtain access to an investigational product to treat their patient. In the particular country that I was working with, it seems as though the hospital and the ethics committee and everything very local can make this determination, and that, like you said, you didn't have to go to EMA in order to get these approvals. So that was the one experience I had. It's my also understanding that the EMA is looking into this a little bit more, and as new things happen, people need to figure out how to regulate it. So I don't know if going forward it's always going to be local review, but it is the case that that is the experience I had with this specific case.
Yeah, and very often these because you mentioned the timeline, so contact the FDA way in advance, but also don't plan a meeting too early because you need the information. So I think I guess it's pretty challenging because you very often for these N-of-1 cases, these diseases are very progressive. So there is kind of a window of opportunity very often, and it also needs time to have some proof of concept data for these compounds. So how to contact the FDA in time, but not being too early and not having any preliminary data.
Yeah, so most of the cases I've been working on have all been central nervous system. They've all been in the division of neurology, and they are aware of this now. And we've been able to make really good relationships with our reviewers over at the US FDA. I think there's a lot of excitement about the science and the ability to potentially treat what used to be an untreatable population. So we've always had really great relationships, and the FDA has always been really supportive. So we have always felt really comfortable saying, hey, we've got this patient. This is what we know right this second. And this is the small amount of natural history data. What can we do? And we've always had a really collaborative relationship to say, okay, well, this experiment is going to read out on this date. You know, it's going to be 20 days before the meeting. Can we give you that data at that point? So just being really open and honest has worked really well for us to navigate a lot of those timing challenges.
Great. There is one question in the chat. If we can just follow up with that now, I think. Have you seen a difference in the regulatory processes since the changes of the NBER?
I am not entirely sure if NBER is an acronym I'm not familiar with, or if it's a typo and should be Center for Biologics CBER at the FDA. And I'm going to answer as if that's the latter part is the question.
Yes, the typo.
Yes. Okay. So these drugs, again, have generally been, these cases have usually been treated with ASOs. Those get treated, those get reviewed at CDER, so the Center for Drug Evaluation and Research, not CBER. I think a lot of what is going on in politics and in the US news and media in our Center for Biologics tends to be around products that we just aren't using. We're not using vaccines and that sort of thing. So fortunately, that doesn't seem to have impacted as much in my experience. I think that we're still, the drug regulations are still where they're at. I think there is a general sense of how could we treat patients better and how can we get rid of red tape, but I don't know that that's actually translated just yet.
Okay, great. All right, so let's switch the mic over to Roger then, and we can get started on learning about that.
Thank you, Erin, and thank you, Ashley. Let me share my screen quickly. There, that looks a little bit more like it.
Perfect.
Thank you. Thanks again for inviting me to this. This is really interesting. I'm Roger Paxton. I'm a research manager for the Precision Medicine Institute at Children's Hospital, Colorado. And I'll be discussing funding individualized therapies. And just to follow up on where Ashley left her bit of the presentation, and to keep everybody's hopes and expectations metered, this is going to be very descriptive and not prescriptive regarding the current state of funding models. So a quick overview of where we'll be going today. We'll be discussing, first of all, the reasoning behind the importance of individualized therapies, thinking through pathways to treatment, and then with that, thinking through aspects that require funding in this process. We'll be discussing historic funding models based on a survey, and then we will have some parting thoughts as well. So individualized therapies, why are they important? Well, first of all, what are they? They are treatments that are tailored to one to few patients, and they're frequently genomically oriented, largely ASOs at present, but we are seeing greater involvement in other therapeutic modalities as well. Now, the need for these targeted treatments. These are meant to treat the rarest of the rare conditions in populations as few as N of 1. And with that in mind, there are typically no existing treatments for these conditions. They're debilitating and they're frequently fatal as well. And it's important to keep in mind that while we might have only one patient that we're treating, it's not only the patient that we are actually treating. These conditions affect families and communities as well. And while these treatments are rare, collectively, all of these rare conditions are actually quite common. I want to thank the authors of the manuscript cited here for this really telling visual with regard to the differences in pathway to treatment in traditional drug approval processes versus N of 1 treatments. So in the upper portion of this figure, we see the traditional process involving candidate discovery and optimization, the phases of clinical trials through which this goes, and then approval to launch. And then in portion B, we see the model leading to an InnovOne treatment with treatment design, proof of concept studies, manufacturing, and safety studies. Then moving immediately into individualized treatment and monitoring. And what we can see here is that, very interestingly, we essentially, in developing and treating with InnovOne, we compress phases 1 through 3, and then post-marketing studies essentially into what we see here in the red, individualized treatment and monitoring. So these are most frequently, by definition, first-in-human studies, and moving through safety and efficacy as well, and then, of course, monitoring and optimization. So this, of course, requires a great deal of resources. And basically, all of the resources that are necessary to bring drugs to bear in traditional development processes are mirrored here. And this is not meant to be all-inclusive. That said, it is meant to sort of shade how much domain expertise is necessary. So thinking through patient identification, needing geneticists, needing treating physicians, candidate therapeutic development, manufacturing, toxicology and safety testing as well. Thinking through outcome measure development, this one is particularly unique and particularly important in these investigations, as we can often tailor outcomes to the individual who is being treated and what is most valuable to them, their family, and their community. Regulatory, thank you again, Ashley, for that, as well as treatment at our institutions. And of course, this involves a great deal of facilities and equipment that are necessary as well. So genomic testing to discern what is the underlying cause of said condition, and then thinking through how that leads into therapeutic development. Toxicology and safety testing once again, manufacturing, and then clinical treatment at the institutions as well. So of course, this involves a great deal of personnel and infrastructure that are required. And note that this slide is only thinking through personnel and infrastructure required at the treating institution. And again, this is not all encompassing. So in terms of personnel, we need principal investigators and knowledgeable clinical teams who have hopefully been involved in research historically and who are familiar with InnovOne clinical trials, or they are in very close contact with folks who are knowledgeable. Our basic and translational research teams, and then leading into our clinical research coordinators and research nurses, the folks who are truly boots on ground implementing this sort of therapeutic. Research protocol specialists who can best design the research protocols to better support the entire clinical and research teams. Regulatory specialists, research pharmacists who are familiar with dealing with therapeutics that are not yet FDA approved, and then social workers as well. So with this, these personnel are best supported by infrastructure, institutional review boards for oversight and ethical and safety considerations, clinical research and trial units, laboratories, and then the all-important legal support as well. So with that, there is one overarching resource that is necessary to support all of these personnel, to support this domain expertise, to support the infrastructure, and that is, of course, funding. So we naturally think, how have these been funded historically? So with that, a little while ago, in collaboration with the InnovOne Collaborative and their members, a survey was sent to constituent members of the InnovOne Collaborative. The goal of this was to think through funding models that have successfully been used for InnovOne clinical trial development and implementation as well. It's important to note, however, a caveat. This was aimed at funding models. This was not aimed at actual funding amounts. And this is due to a high degree of anticipated variability by institution, geographic region, and time, as well as there for any one of these InnovOne clinical trials, most of them not having a single source of truth across the development stages. So for example, one group might have very good eyes on the manufacturing cost, well as that same group might not have a great deal of insight into institutional onboarding costs, etc. So with this, we broke this down into a variety of buckets from which funding might come for these InnovOne therapeutics. It's included government, institutional, and by institution, I mean treating institution, private donors, industry, nonprofit, insurance, and then of course the catch-all other. So we'll be looking at the results of this survey in a variety of ways, starting with this table. To orient ourselves to this, the leftmost column, we have our funding source. The center column, how many clinical trials that we investigated utilized each of these buckets, and then the proportion of the funds coming from each of these in mean. It should be noted as well that our sample size ended up being 20 respondents to this from unique clinical trials. We can see here in very quick strokes that there was very, very few that actually used government funding and industry funding, but we can certainly see that the majority of these utilized funding from the treating institutions as well as nonprofit organizations as well. We can see these broken down in a slightly different way here. So on our x-axis, we have, again, our funding sources. And on our y-axis, we see funding percentage. Each of these dots represents a proportion of a single investigation and where they fell in terms of funding proportion. And what we can see here, again, in broad strokes, is an extreme degree of variability, this looking much like essentially a shotgun approach to funding. But we can see, again, that the majority of studies did use nonprofit and institutional funding. Now, looking at this once again in a slightly different way, on the x-axis, we have our discrete clinical trials broken down, all 20 of them. On our y-axis, we see funding percentage that each of these clinical trials utilized. And then on our legend on the right, we can see color-coded what these funding sources were. Again, we can see an extreme degree of variability, but what we can see, once again, is strong purple and a strong orange in this, representing both, again, the nonprofit and institution funding sources. So what are our takeaways from this? We can see that there is at present no common funding model for individualized therapeutics. We can see that institutional and nonprofit resources are frequently utilized, and we can see that there is a need for a well-understood common approach to resourcing that we can use with great replicability for the sustainability of these sorts of therapeutics. So a couple thoughts, again, not meant to be overarching in any way, about potential sources of funding moving forward. So one would be if we could find a path forward for greater government involvement, and then advocating for allocation of resources for this as well. So just one example of this, in 2019, the NIH dedicated $5.74 billion to oncology research, while dedicating $38 million to rare disease research. With just the numbers of patients impacted by rare disease, I think we could reasonably advocate for greater amounts of funding with this in mind. Another that we have used to date consistently are nonprofits, and it is commendable, the work that they have done to bring these treatments to those patients in need. But there are ethical questions that have been raised regarding participant and population selection for treatment. And then, of course, with nonprofits as well, concerns regarding limited expected scalability of funding per organization, and thinking through that in terms of, once again, sustainability. There is hope, however, and we see great ongoing effort. First of all, in rare disease advocacy. So rare disease organizations, patient and family groups, and the scientific community at large. We also see great work ongoing in scientific understanding, and shared learnings regarding safety and efficacy these therapeutics. And it's important to note as well that with these greater understandings and the greater ability to develop and manufacture these treatments, the issues that we face are really rarely scientific anymore. These are really issues of infrastructure. So thinking through regulatory funding as far as a variety of other issues, those are really where we see the greatest issues at present. And also greater work by nonprofit partners for these, and hopefully greater groups coming on board as well. So thinking through in LoRa, for example, developing, manufacturing, and providing ASO treatments for these InnovOne conditions. And then groups like the InnovOne Collaborative as well, with their donated resource center, where a great amount of very, very kind organizations have actually donated resources in kind to support development and treatment with individualized therapeutics. And then with promise as well, a story came out in Endpoints that Prasad of the FDA has stated that there will be an approval pathway for InnovOne therapies coming soon. We shall see, but we shall also hope for this. I can say that there is another approval pathway that has recently been announced by the FDA, an approval pathway for therapeutics treating rare disorders. Their definition of rare at this point is 1,000 or fewer patients in the U.S., where they are asking for a pathway to approval with one very robust, well-designed study, and then a great degree of supporting documentation as well. I don't pretend to be well read on that at this point, but it is promising moving forward as well. So with that, I would like to thank all the thought partners involved in this process. This has truly been a team effort and a team sport in thinking through this information, collecting the information, and then collectively thinking about how we can use it as well. So most of these folks are members of the InnovOne Collaborative. So I would like to thank all of them at this time. And thanks again for the venue today.
So once again, so if you have questions for Roger, please use the Q&A function. So it's a bit challenging, right? So I was looking at your funding bars, which color mostly had the colors of the non-profit organizations, and so how to change this? How to get more funding from the government?
Yeah,
because
so
I think what you stress is that there are lots and lots of people are affected with these rare disorders, and it's so not so how to raise attention for these kind of mechanisms.
Yeah, so that I think is an excellent question, and once again comes back to advocacy as well. So our ability to treat patients with extremely rare disorders to the tune of InnovOne, that's very recent, right? I mean, it just in the last 10 years have we even had the ability to conceptualize that. So I think raising awareness that we do have these abilities now, and getting folks in the government and in the community to better understand that now that we have these capabilities, we actually have the platform in which we can treat a great many individuals, even though they do have different rare diseases. So I think a lot of it has to do with advocacy and awareness and communication, not only by our patient and family groups, but also greater communication coming from the scientific community and treating physicians as well.
And if I can just add maybe a little bit more of a prescriptive, what I would like the government to do comment. A lot of the federal grant RFAs don't apply here. You're looking for, they want to fund clinical trials of multiple subjects, and it's just, I would like them to put more RFAs out for N of 1. And second to that, I'm sure everyone on this panel can tell you how long before you actually get the money in hand. I mean, our patients can't wait for a review cycle and then the contracting, and then someday next year you're going to get a couple of dollars. Like the current structure for US federal grants doesn't support this either. There needs to be a shift in how they're funded and what they're looking for as well.
And then the other way around. So let's suppose we get the health insurance, and the health insurance will cover all these therapies. But yeah, I think what we see in the last few years is that the number of individual therapies is raising quite. It's getting quite a big jump. So one might expect to have many of these therapies out there in the near future. So how can we pay this then as a society without having like a reasonable amount to pay for the health insurance costs?
I think health insurance is also a little bit tricky because they'll pay for the drug vial and the injection and the MRI and the sedation, and so, but you're not going to get an insurance company that's going to pay for your rat study. So I think, as Roger's slides showed, I think you're going to have to find a lot of different funding sources for a lot of the different parts along the way.
I think that one element to that as well, and this is very crystal ball gazing, so I could be completely off base on this. But if we were to land in a space where there was some form of FDA approval for these InnovOne therapeutics, then that would allow institutions to actually bill payers for this. And at that point in time, a manufacturer might be able to set a price point where they might actually be able to gain some degree of funding for the work that had been done leading up to treatment. Again, I'm not an expert in that area, but it is something that we have discussed as a possibility in the future.
Yeah, because yesterday I was at a conference, and there was actually also a talk about this InnovOne, and then they said that the estimated cost just for an academic to do everything in academics is like approximately a few €100,000. So that would be just to cover someone doing the experiments, the natural history study, do the tox study on the drugs, these kind of things. So that would be quite costly actually for insurance companies.
That's absolutely true, and I think that a few €100,000 that feels a little bit conservative. Even I would anticipate that it costs more than that, though again don't have the hard numbers to back that up. I think on top of that as well, thinking through insurance and payer support of this sort of thing is important to think about as well institutionally, particularly with ASOs. If these treatments prove to be safe and efficacious, then we're going to have to be treating these patients for life. They're going to be needing dosing for the rest of their lives, which, like one dose, like that might not be that costly, but thinking through doing this over the course of years, and then to your point as well, the fact that we anticipate that we will be seeing more and more of these sorts of treatments in the future, that begins to really accrue cost over time for treating institutions.
Yeah, that brings me also to the following point. I don't want to be so I really like how the field is moving forward. But so are people also thinking on how to improve the infrastructure? Now we are talking about a few dozen cases, but like as before, we can easily scale this up to hundreds, thousands, 10,000s of cases of individuals that can be treated with these kind of therapies. So how to build the infrastructure, because these individuals need to be monitored in the hospital. They need to have these monthly, bi-monthly, 3-monthly injections. They need to be followed up. If you want to check whether the treatment is working, natural history studies need to be done. Also, the validation studies need to be done. So, are you also working on or are people working on the implementation side?
I think Ashley and I can both probably speak to that a little bit, but that being one of again, sort of another plug for the InnovOne Collaborative and the work ongoing with that group, thinking through not only resources that can be provided to individuals, but also acting as sort of a point of reference. So, for example, thinking through data safety monitoring boards that have been historically used, that are familiar with InnovOne clinical trials and the specific care that is necessary there. So yeah, there is work going on. Much of it, quite frankly, has yet to be matured, but efforts are ongoing.
Ronald, you're muted.
Thank you, Erin. Classic. Okay. Question remark from Felix. So, did you manage to get a detailed cost breakdown? So, the clinical care, manufacturing cost, drug development, the regulatory cost of the various steps of these different groups. And I think there's also a paper cited from Tim Yu, somewhere in the 1.4 to 1.2 million range. So can you tell something about the breakdown of these costs?
So sadly, no, that was not included in the survey. So we were very cognizant of the individuals to whom we were sending this survey, and so we were very focused in on one specific question that was the essentially buckets of funding. But that is an excellent question that I would love to have addressed in the future. And I have seen Tim's work, and that cost estimate rings, I think, it rings true to me. That I don't have great data to support it, but it feels right as far as the gut instinct.
Yeah, it might actually be quite a nice follow-up question to ask those people. Okay, do you have any idea how to split the cost before moving to these? Because one might also expect that many of these costs are not even included in this budget.
Yeah, that's true. So I wholeheartedly agree. That's a great question and would be an outstanding follow-up as well.
Okay, question from it was jumping. Question from Jin. So if you can create a N-of-1 therapeutic development pipeline where one could develop multiple similar types of therapeutics in parallel, would funders be able to fund the pipeline rather than an individual treatment? So it's like more a broader approach.
I love that thought, and I think that would be aspirationally a hope and a goal. Any thoughts from you on that, Ashley?
Yeah, I think we're going to need, at least in the US, a little bit of regulatory flexibility on that. Currently, and I'm not going to pretend to know too much about the science. I'm not scientifically trained, but despite each ASO being generated by different nucleotide bases, the FDA considers each one a new molecular entity, and they currently require toxicology for each one. So if we can get some sort of agreement that, if we're using the same backbone chemistry, if we're using, certain sorts of, if there's a way to get them to agree that we can do this sort of platform-like or pipeline-like, we could maybe trim costs. But currently we're operating in an environment where each one is an entirely new product and needs its own supporting data.
And so I agree. And as a follow-up to that, I've had line of sight to the approach to FDA's requirements for toxicology reporting on or testing on a variety of these, and they look sometimes strikingly different. So with that in mind, that can drive a differential in costs, but also that further demonstrates how they are treating each one of these as its own therapeutic agent.
Yeah, I can imagine it's very difficult, and it should be really on a case-to-case situation. It should be judged, I can imagine. So, in the chat, I see that Felix is happy to participate in a follow-up questionnaire. So I think write down his email address and contact him.
Awesome. I greatly appreciate that. Erin or Ronald, would one of you mind sending me his contact information?
Yeah, I will. It might be that we have it. And Felix, if you are listening, can you maybe please send your contact details to info@oligotherapeutics.com? Ah, it's there.
Oh, it's right there.
It's
Perfect.
in the chat. Also for the other listeners, also if you don't see this live, but when you watch back this webinar, feel free to send your contact details to info@oligotherapeutics.com. I think we can also put our email address in the chat.
Tossing mine in the chat at present.
Yep. And then in the meantime, we have a question from Hillary. Good point. The chronic usage. How does the trial pan out after a decade? Can the trial costs come down, or does the intensive hospital monitoring continue, or is the long-term planning for these trials something that will follow later when we get there? So anything about all the follow-ups and the treatment. So what is actually the longest treatment that we do have now for an N-of-1 trial? So how long is this ongoing?
I think that's one at our institution, and it's coming up on, let's see, December of 2019. So we're coming up on 6 years. And, you know, we're currently at a stage where the patient can only come in, it needs to only come in for the day, and it only needs to be, doesn't need to be monitored overnight, but there's still the costs associated with doing the injections. We're still doing serial MRIs. We're still doing all sorts of assessments. So, you know, we have moved away from, we're at this, you know, I couldn't comment on the actual interval, but, you know, 2 to 4 months between doses. So we're doing, you know, three to 5 of these a year, which is nice, other than the escalation that ramps up at the beginning, and we're not doing significant monitoring, but these are not oral meds that you can just send home. So they're just inherently more expensive to treat. So that and hospital prices have certainly gone up in the last 6 years. So I think there are some things that we've been able to strip away, but some things just get more expensive over time. So I don't have a significant price drop at this point.
And I would echo all of that from what I'm seeing at Children's Colorado as well.
Yeah, because I was also thinking, I never actually heard of that. So do people also investigate, so how long you can store these therapeutics? Because, you know, I'm not sure whether you can manufacture one batch and can use this for a lifelong treatment, or is this only, can you store this for only 10 years, and do you need to make a new batch after 10 years? Is there any information on that?
So we, in the very broad sense, did the calculations on how much drug you would need to treat for decades. So when we had our initial batch done, we have a lot. We do have to do stability testing every year to ensure that it is still stable and pure and that sort of thing. We have also done some work in potentially treating other patients with this specific mutation. I mean, the more and more people are identified with certain genetic mutations over time. So I don't think that our one supply will last a lifetime. I think there are calculations that people do to try to do that, but you also want to balance, is the drug you're injecting 30 years from now still good? So we definitely don't want to be manufacturing every year, but I don't know. As Roger said, you know, that we've been doing this for a while, but not that long of a while. So I think there's going to be a balance between manufacturing something for someone's whole life versus what is the right. Do we want to plan for 10 years? Do we want to plan for 20 years? I don't have the scientific knowledge to answer that question.
Can I ask a N1C question? So I think when I saw when I go to the website, I see that people can also be part of the N1C community. So how to become part of the community, and what are you looking for? Are you looking for some specific experts on a particular disease, or clinicians, or more on molecular scientists, or so can you elaborate a bit on that?
We'll take everybody.
Everybody.
Yeah.
That's good.
Yeah. I mean, this is
response.
they're between the geneticists that are trying to identify patients, the people that are doing ASO screening, the people that are working on cleaner, safer chemistries, the regulatory people like myself, the clinicians that have to figure out there's only 10 people in the world with this disease. How do we know if they're getting better? I mean, we are trying to make an impact all the way along the pipeline. So anything that anyone has interest in or experience in, we are looking for that. If you go on the website, you'll see all of the different work groups that are available to try to bring like-minded people together, whether it's regulatory people, chemistry people. You know, there are a lot of different ways to get involved with either just listening and contributing in our seminars, participating in our work groups, and then also becoming an N1C member to have a little bit more access to some of the content, our annual meeting that is happening in October, and more. There's also some different communications platforms to foster communication between all of our different stakeholders.
Yeah, and I think you probably also have a booth during the OTS.
Yes.
So they can visit you guys, and they also have very nice goodies. So you can go there for a chat, and you can also pick up a nice goodie.
I would welcome anyone from this seminar and beyond to please do come visit us at the booth.
Let's see, Erin, do you have any other questions?
Yeah, that was great. I mean, people never know where to start, and so it's great to have this launching off pad. It's great to learn more about the resources and what's going on, the mission of the N1C. And so we're really excited to connect again in Budapest. If anyone is interested in the meeting information, I put it in the chat. You can also reach out through info@legaltherapeutics.com, and I'll get routed back to Ronald or I will share it with you. Roger shared his contact information as well, so he's a great resource. And I think, Ashley, you're on LinkedIn, right? You're both on LinkedIn, so people can reach out as they wish.
Yes, and I'm also putting my email in the chat.
Oh, perfect. Great. So take note of those emails. We have a few webinar sessions coming up. We have Professor Maureen McKeag will talk in late November, and we may have another early November session, and of course we have the meeting in October. So this is the last time we'll see you until we can see you in person. So we want to thank everyone, Roger, Ashley, again, for giving such a great session, and we can wrap things up there.